we start the Krebs cycle. A little bit of history here. Krebs cycle, or the citric acid cycle, they're the same thing. So Krebs cycle, citric acid cycle, same thing. Was named after Hans Krebs, who worked out the pathway, won the Nobel Prize. We talked about Nobel Prizes. Won the Nobel Prize in the 1930s. Never was comfortable with it being called the Krebs cycle, so he preferred the citric acid cycle, which is indeed what he called it, the citric acid cycle. Some call it TCA, which is the citric acid cycle, TCA cycle, or the citric acid cycle. Okay? But it completes the oxidation of molecules and generates NADH and also FADH in the process. Now, I'm going to put a little asterisk out here because for the test, I'd really like you to take a look at this. I'm going to use that color, I think. To really take a look at this particular picture, this figure from your book. It's really good. It's really good. Remember that we've got 2 acetyl coenzyme A molecules. So everything in here is double, okay? Or another way to say it is, 2 times. Okay, 2 times here, all right? So here, for instance, you see we're forming 3 NADHs, okay? And then you can see here we're forming ATP from the addition of a phosphate group to a ADP in that process. In here, we're forming FADH2, which, if you think back, that's another coenzyme we talked about earlier, okay, or another electron transporter, if you will, okay. Now, everything here is multiplied by 2 because we have 2 acetyl coenzyme A's, all right. So, just kind of to total this up, just in the citric acid cycle, okay, I'm going to put TCA for short, citric acid cycle. But what you have here is 3 times 2, or what we have is 6 NADH plus H plus, okay? So we have 6 of those, all right? Then what we have is 2, let me back up here. You've got 1 times 2, or 2 ATP in that process. Okay, that's this down here. And then finally, what you have is 1 times 2 equals 2 FADH2s. Okay? So our net here is going to be 6 NADPH, 6 NADH plus H plus. You're going to have 2 ATP, and you're going to have 2 FADH2. Okay? Now, if you think back for just a second, over here, what I had you write in the margins, right, was that you actually had 4 NADH plus H plus. So if we add the 4 to the 6 from the citric acid cycle, okay, you're going to wind up with 10 NADH plus H plus. Okay, so that's just from the previous screen. Here again, if you keep writing these in your margins, the math will be a little bit easier as we go along, okay? But I just want to point out that the total here would be 10. The 4 from the oxidation of pyruvate and glycolysis, and then the 6 just from the citric acid cycle. So that gives us a sum of 10. But let's continue here, okay? So this is a really good figure for you. Really good. All right, I'm going to give it a star here, asterisk. I look at this because it helps to summarize the total. All right. Now, I will show you the citric acid cycle, but here again, don't panic. Okay, I don't expect you to know all these intermediates, but that's what I'll show you. Okay, so this is a good slide. This is going to help to summarize. So, here again, I'll put an asterisk by this one too. During the citric acid cycle, right, the 2-carbon group, the acetyl coenzyme A, is joined by a 4-carbon group. Citrate is degraded back into what we see as a 4-carbon compound. 2 CO2s are released. 1 ATP is produced. This is just a summary here of the previous slide. During the citric acid cycle, right, the 2-carbon group, the acetyl coenzyme A, is joined by a 4-carbon group. Citrate is degraded back into what we see as a 4-carbon compound. 2 CO2s are released. 1 ATP is produced. This is just a summary here of the previous slide. Okay, 3 NADH, 1 FADH, okay, are produced. Okay, here again, that's what I want you to remember. This is what I want you to remember for the test. All right. Remember, there are 2 molecules of acetyl coenzyme A for each initial glucose. Therefore, there are 2 turns. I like that so much, I'm going to highlight it here. 2 turns of the citric acid cycle, okay, per glucose molecule. So what does that mean? Well, just this, okay? Everything is doubled in the previous statement there. So, you know, where we have up here 1 ATP, one ATP, okay, we double it, so now we have 2. Where we have 3 NADH, we double it, so we have 6. Where we have 1 FADH, we have 2. Okay? And then this is really key down here, so I'll put an asterisk by it. Okay? When we sum all these together, take into consideration glycolysis, the citric acid cycle, everything together, what we wind up with as a sum total is 4 ATP, okay, 2 from glycolysis, and then 2 from the citric acid cycle, 10 NADH, that's what I was illustrating on the previous slide, and 2 FADH2s. Okay? So the energy is banked in NADH and FADH2. Now, I want you to kind of write out to the side here in your notes. NADH, okay, equals, for each NADH that you get, you're going to wind up with 3 ATP. I'll explain this later with the oxidative phosphorylation. For FADH2, okay, you're going to wind up with 2 ATP. All right? So if we do math here, all right, we have 10. So if I go 10 times 3, it's going to give me 30 ATP. And if I go 2 times 2, that's going to give me 4 ATP. All right? So I'll explain this when we get to oxidative phosphorylation. But just remember that for right now. Okay. So this is the citric acid cycle, and I'm not going to go through, I don't expect you to know all these intermediates, so I'm not going to spend a whole lot of time on this. But you can see the reduction reactions going on here. NAD is reduced. There's 3 of them. NAD is reduced again here, okay, and then it's reduced again here. So there's your 3 NADH plus H plus. I mean, it really is an amazing process, and you can see why, for instance, he won the Nobel Prize for this particular process. We're working out the process. Here you can see FAD becomes reduced, and you get your FADH2, okay? And here, for instance, you can see the production of ATP, okay? And remember, again, this happens twice, so those are really kind of the key steps that I want you to see. Here's citrate right here, and this becomes what we see as citric acid. So this is where the name citric acid cycle comes from in that particular process. All right. Don't panic, okay? Just wanted to show you what the citric acid cycle looks like. All right. So we're in the home stretch now. The final stage of cellular respiration is oxidative phosphorylation, which involves the transport of electrons and also chemiosmosis, which I'll explain. But the process makes it possible to create a hydrogen ion concentration gradient across the membrane, and then to use that energy in the gradient to drive ATP synthesis. So here again, you're going from a high to low situation here, high to low situation. Okay, the electrons from NADH and FADH travel down the electron transport chain, and then again, oxygen is the final electron acceptor. I'm going to star that. Okay, I'm going to highlight that. All right, oxygen, final electron acceptor. Oxygen picks up the hydrogen ions and it forms water in that particular process. Okay, energy is released and used to pump hydrogen ions from the mitochondrial matrix to the intermembrane space, and it's like the matrix. Okay, I can't tell you about the matrix. I have to show it to you. All right, all kidding aside, the intermembrane space is between, for instance, the matrix and then the outer membrane of the mitochondria, because you have a double membrane, like the nucleus of the cell. You have this double membrane. So I will show that to you. But for right now, the key point here is that it pumps hydrogen ions to this intermembrane space. The final part is this chemiosmosis. As those hydrogen ions diffuse back now, going down their concentration gradient, here again, high to low, they go through a pump that's called ATP synthase. It's actually an enzyme. How can you tell? Ends in ASE, okay? And that actually drives the synthesis of ATP. So that's it. Let's look at these last steps here, shall we? All right, here we go. So, let's get our orientation. This is the outer mitochondrial membrane. I'm going to change colors here again. This is the outer mitochondrial membrane. Okay. I'll do that a little bit better. All right. Outer mitochondrial membrane. This right here is the inner mitochondrial membrane, right here. All right. And integrated into it, you have these proton pumps. Okay? And you can see here's hydrogen, 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 like that. Okay? Now, earlier, and then finally, this is the matrix. Okay? This is the mitochondrial matrix. So you've got kind of a layer in between, right, like this. Okay? This is the intermembrane space, right here, intermembrane space. Okay? This is getting really kind of cluttered here, so I'm going to erase some of this here. All right. Now, earlier, I said to you that NADH plus H plus, plus H plus, okay, was equal to 3 ATP. You might recall that. And I said FADH, okay, 2, was equal to how many? 2 ATP, okay? Now, this is where it comes from. Watch closely, okay? Clean this all up here. It's all up. All right, so here's where it comes from. In the case of NADH, starts here. It starts with the very first hydrogen pump, or the very first proton pump, okay? And so NADH takes those 2 electrons. Remember, it's an electron transporter. Okay, it's a coenzyme, if you will. It takes those first 2 electrons, transports them to the very first of the proton pumps. Okay? Then it goes to the second, like so. Then it goes to the third, like so. So you're transporting 3 hydrogens in that process. Okay? Now, the difference that we see with FADH, okay, if we look at that for just a second, the difference with FADH down here is that FADH, okay, it starts here really with the second proton pump. So you get 2 hydrogens in that process. Okay, and so that's why you get 2 ATP ultimately. Alongside the short story is ultimately you get 2 ATP from FADH, whereas you get 3 from NADH in that process. Okay, and then the other thing to point out down here, okay, is that in either case, waiting at the end of this whole thing is oxygen. Oxygen becomes the final electron acceptor. Here's my final 2 electrons right there. Okay, it accepts those 2 electrons, and you guessed it, we get water in that process. Okay. Now, what happens to all these hydrogens? Okay. Well, all these hydrogens over here, they form this gradient, if you will. Let's go to maybe blue or gray. That's good. Okay. So they form this gradient, if you will, and over here, of hydrogen ions. Okay, so there's a gradient over here. And they start moving down through this enzyme called ATP synthase. Okay. For every hydrogen that moves down through ATP synthase, it supplies the energy to combine a phosphate, right, with a diphosphate. So what happens when you take 2 and combine it with 1? You come up with a triphosphate. Okay? So what does that mean? Well, what that means is that since you get 3 hydrogens now, 3 hydrogens for every NADH, ultimately, you know, these 3 hydrogens move down. Ultimately, what you wind up with is 3 ATP. Since you get 2, okay, hydrogens, 2 hydrogens now, for every FADH, 1, 2, okay, what that means is that you wind up with 2 ATP in that process, because it supplies just enough energy. And here again, you're going from high to low, high to low. Okay. All right. This is just a picture, just kind of showing you here as the hydrogen moves down through. Oops. As that hydrogen moves down through. Oops. You can see it right here. It's moving this ATP synthase. It's like a rotor. I'm not so concerned that you know this catalytic knob, but what I am concerned that you understand is that it supplies the energy. The hydrogen moving down supplies the energy to combine a phosphate with a diphosphate and form ATP in the process. So now, at the end here, let's do a summary, and let's talk about glycolysis. Glycolysis, what do we learn? Well, we learned that we had, 4 ATP, right? We used 2, right? So we wound up with 2 in the process. Okay. We learned that we generated, pyruvate. Okay. And we generated 2 NADH. Plus H plus. Okay? That's glycolysis. What do we learn about pyruvate oxidation? Okay. What we learned was that we generated in the process 2 more NADH plus H plus. Okay. That we generated, 2 acetyl coenzyme A's in that process. Okay, that's what we learned from pyruvate oxidation. What did we learn from the citric acid cycle? Okay. Well, we learned from this that we generated 6 NADH plus H plus. Okay, we learned that we generated 2 FADH. Okay, and we learned that we generated 2 ATP. All right. So, in summary, when we add this together, what do we wind up with? 2 and 2. Excuse me. 2 and 2 and 6. 2 is 6 plus 2 plus 2 gives us 10 NADH plus H plus, like that. Okay. The 2 FADH, like that. Okay. And then we wind up with 2 ATP here, 2 ATP there, so we wind up with 4 ATP in that process. Okay. Now, through this very last step here, I'm going to put down, I like oxidative phosphorylation, so I'm going to put that. You can also call it electron transport chain. Is perfectly correct. You can also call it the chemiosmotic mechanism. But I like oxidative phosphorylation. So what do we get here? Well, we've got 10 NADH plus H plus. Okay, that's going to give us 30 ATP. All right, we've got 2 FADH. That's going to give us 4 ATP. Okay, so that's going to give us a total of 34 ATP. I can write that better. It's a little hard to write with this pen. 34 ATP. Okay, plus the 4 up here. Okay, gives us 38 ATP total. Okay, so that's what we wind up with. That's, our net, if you will, at the end. Thank you for watching this presentation. You might